Have you ever had to hand in an assignment and noticed a spelling mistake while checking it? This is when you go back and, as easy as it is, highlight the error to either correct it or delete it. Now, imagine if you could do the exact same thing but in the genes making up your DNA. What would you think if I told you this could be possible? Yep, that's right. It is actually now possible to modify human DNA thanks to a genetic technology called CRISPR Cas9. Okay, so originally CRISPR was the name given to some bacteria's immune system. A study published in 1987 showed how some bacteria were able to defend themselves from viruses by destroying their genetic material. It was discovered that the bacteria's DNA contained a series of identical sequences that repeated themselves, that were interspaced by other segments that, on the other hand, were unique and even contained fragments of viral DNA. But where did these foreign DNA segments came from? If the bacteria manages to survive the viral infection, it captures a fragment of this viral DNA and integrates it in its own DNA, in the spaced DNA regions I mentioned before. These make up the clustered, regularly interspaced, short palindromic repeats. This way, if the virus tries to reinfect in the future, the bacteria will transcribe the viral DNA into RNA along with the nuclease enzyme Cas9, which will guide the RNA strand along the bacteria's genetic code looking for a complementary strand, successfully recognizing and destroying the virus by specific site cleavage before the infection even begins. CRISPR was implemented on the lab in 2012 by Jennifer Doudna and Emmanuel Charpentier while studying the bacteria Streptococcus pyogenes, turning it into a revolutionary genetic engineering technique. They realized that if a specific RNA was constructed for a gene of interest and engineered into the Cas9 protein instead of viral DNA, the CRISPR technique could in fact be used to engineer any DNA sequence at a precise location. This specific RNA sequence is now known as guide RNA. Once inside the nucleus of the cell, the guide RNA Cas9 complex will lock into a short sequence known as the protospacer adjacent motif. This is when the Cas9 will unzip its DNA and match it to its target RNA, then cleave it, resulting in its gene inactivation. If a new piece of DNA carrying a desired sequence is inserted, it will pair up with the cut ends to replace the original sequence, achieving a successful gene modification. This method of gene modification is currently being studied as it is believed to have the potential to revolutionize medicine to fight disease, cure cancer, or even improve human species. This paper talks about a study about the implementation of CRISPR-Cas9 system in the research and treatment of cardiovascular diseases such as atherosclerosis, hypertrophic cardiomyopathy, congenital heart disease, and others. So you must be thinking, if this is such a revolutionary technique that could potentially eradicate terminal diseases and human species, then why have we not taken it a step ahead? Well, the truth is there are some ethical concerns hiding behind this topic. First of all, we agree that CRISPR would be a fantastic approach to the treatment of multiple diseases, right? The problem comes when considering which genetic conditions need fixing. Is Down syndrome a disease? Is dwarfism a disease? Well, some may think it is. Many in the dwarfism community may disagree. Are these actually diseases, or is it a way of discrimination from society towards different communities of people? Furthermore, the line between therapy and enhancement has become more fuzzy over time, even for diseases we agree on. For example, in Alzheimer's disease, depending on which variant of the APOE gene an individual has, it increases, decreases, or doesn't affect at all on the risk of developing the disease during the later stages of life. Imagine if an embryo was genetically engineered to switch a high risk for a low risk variant. Would this be a therapy or enhancement? Unlike this hypothetical case, there are millions of genes linked to lower risk of heart disease, diabetes, HIV immunity, all the way to bigger muscles, eye color, less body odor. Where would you draw the line? Is it ethical to genetically engineer an embryo to have blue eyes, light hair, and lower body fat percentage as if it was a puppet design? We must also take into consideration the fact that when we genetically modify an embryo, we are talking about germline editing, which differs from somatic editing. In other words, unlike somatic editing, germline editing involves sperm, eggs, and embryos, meaning that whatever change we make in the DNA will be passed down to future generations. Eventually, human species will be forever marked with a signature of gene editing. Where would the planet's natural biodiversity stand then? Shouldn't we be protecting it instead of altering it? Do we have the right to play God, as some would say? Some alterations made to the human species could potentially lead to mutations that will destroy or permanently alter our own species. And you, what do you think of the CRISPR system? Do you think that the possible benefits overcome the clear downsides and ethical concerns, or should we leave nature do its job?